Let's say we have DNA with a coding sequence shown here. When we transcribe this DNA sequence into RNA, all we need to do is copy the sequence down, remembering to replace the thymines with uracils, since RNA has uracil instead of thymines, but essentially you copy the sequence of the coding strand down. And that is why this process is known as transcription, which is you make a literal copy of the DNA. On the other hand, if during translation we make a polypeptide based on this messenger RNA sequence, then we end up with a sequence of amino acids: methionine, threonine, tyrosine. And therefore, there is not a direct correspondence between the letters of the messenger RNA and the letters of the amino acid sequence, and there must be a code involved in translation, a code that tells us how one letter or some combination of letters in RNA correspond to one letter in the protein. And the first question one could ask is this an overlapping code or is this a non-overlapping code? In an overlapping code, one would use overlapping sets of letters to specify proteins. For example, if we had the transcript as written above, AUG, ACG, UAU, UAA, then the first amino acid would be read as AUG. So that's amino acid 1. And then the second amino acid would be read by an overlapping set of bases, UGA. And the third amino acid would be read by GAC, and so on and so forth, so that one amino acid and the next share two bases, and the next amino acid is read by just shifting the frame by one amino acid to the right. On the other hand, if we are talking about a non-overlapping code, then the same transcript might be read in this manner. Maybe if we had one amino acid being specified by 4 base pairs, then this would be the first amino acid, then CGUA would be the second amino acid, and then UUAA would be the third amino acid, and so on. The first one is an overlapping code. And the second case we discussed is the non-overlapping code. Now we know that the genetic code is not overlapping, but how is it that we know that the genetic code is non-overlapping? And evidence that the genetic code is non-overlapping comes from the analysis of a type of mutation in which one of the nucleotides is changed to a different one. In other words, substitution mutations. So let's say this G has been mutated to a C. Now, if in the situation where the genetic code is overlapping, amino acid 1 is AUG and will now be AUC. So you will get a different amino acid. Amino acid 2 was UGA and after the mutation becomes UCA. Therefore, amino acid 2 will also be changed. And the third amino acid was GAC, which will now become CAC. Therefore, amino acid 3 will also be mutated or changed. In the scenario that the genetic code is non-overlapping, amino acid 1, and in this case we imagine that the genetic code is 4 letters, AUGA becomes AUCA, and so amino acid 1 is mutated or changed. Amino acid 2 is CGUA and is not changed because the substitution only happened at the G in the first amino acid, and amino acid 3 is also not changed, and so on. So there are distinct predictions of what would happen with substitution mutations depending on whether you have an overlapping code or a non-overlapping code. And in the case of an overlapping code, you would expect more than one amino acid would be changed when one nucleotide is substituted for another. Whereas in the non-overlapping case, you expect only one amino acid to be affected, which is in fact known to be true, that when you have substitution mutations, only one amino acid is changed, thus establishing that the genetic code is non-overlapping. The next question is, how long is the genetic code? In other words, how many nucleotides specify one amino acid? Now, it turns out that the fact that there are 20 amino acids means that the codon, or the number of base pairs that specify one amino acid, is at least 3 nucleotides long. So let's say that one codon is 1 base pair long. And so we say here that 1 codon is equal to 1 nucleotide or 1 base pair. In that case, if we have a message like so, at every position in the message, we have 4 possibilities: A, T, G, or C. And therefore, since 1 base pair is equal to 1 codon, the polypeptide, or the protein, will also have at every position one of 4 possibilities: amino acid 1, amino acid 2, amino acid 3, or amino acid 4. That is to say that a code in which 1 base pair specifies 1 amino acid can only specify 4 amino acids. And that clearly is not enough, since the code must be capable of specifying at least 20 amino acids. So what happens if 1 codon is 2 base pairs or 2 nucleotides? In that case, if we have a message of this sort, then the polypeptide will be like this. Now, there are in the message 4 possibilities in the first position, and there's another 4 possibilities in the second position, and that means in each position of the polypeptide there are 16 amino acids that are possible. And that again is clearly not enough to specify 20 amino acids. So let's consider the situation where one codon is 3 base pairs. Now, our message will be read in 3s. So every 3 base pairs gives 1 amino acid in the polypeptide. In this case, we have 4 possibilities ATGC at each position, but there are 3 positions, and so we have 4 times 4 times 4, or 64 possibilities, or 64 amino acids that are possible. And 64 is clearly greater than 20, implying that the codon must be at least 3 base pairs long. Now note, all we have shown is that the codon must be at least 3 base pairs. It could be more. It's certainly possible that a codon is 4 base pairs or 5 base pairs. However, this analysis of evaluating how many amino acids 1 base pair codon or 2 base pair codon or 3 base pair codon could code for shows that one codon must be at least 3 base pairs long. So how is it that we know that the codon is exactly 3 base pairs? And evidence that this is the case came from Francis Crick's experiments with what are known as R2 mutants of the T4 phage. Now, the T4 phage is a virus that infects E. coli, and normal T4 phages can infect B or K strains of E. coli. However, the R2 mutants lose the ability to infect the K strain, and therefore they infect only the B strain of E. coli. Finally, also known were suppressors of R2 mutants that reverted the phage so it could infect the K strain again. to recap, wild type T4 phage can infect both B and K strains of E. coli. Crick and colleagues knew of mutants known as R2 mutants that lose the ability to infect the K strain, and so they infect only the B strain of E. coli. They also knew of suppressors of R2 mutations, so a double mutant, where the double mutant was reverted to the wild type phenotype so that it could infect the K strain again. Now, the analysis of these mutations showed that these R2 mutants were either insertions or deletions in the relevant genes. So, for example, if we have a wild type message like so, it would code for, and assuming that a codon is 3 base pairs, histidines. Now, the R2A mutant is a insertion of A at position 4, then the message would become CAU, and then we have an insertion of A, and then we will continue the rest of the message. So you will get CA, then UCA, 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 and so on. And so now, assuming a triplet genetic code, we will get a histidine and then a threonine and the rest of them will be serines. And this is called a frame shift. When the reading frame has shifted so that not only the amino acid with the mutation is different, but all subsequent amino acids have changed as well. Now, the suppressor of this mutant was known to be a deletion. And so let's say we delete the A at position 9. Now our message becomes CAU. We have the first mutation, the insertion of the A, then CA, and then we have U, C, and the second mutation, which is a deletion of this A, and then the message continues U, C, AU, CAU, and so on. This will result in a polypeptide that's histidine, threonine, serine, with a reversion to histidine starting at the 4th amino acid. And this means that except for 2 amino acids, the rest of the polypeptide would revert back to its original composition and result in a reversion to the wild type phenotype or suppression. Another class of mutations known to Crick and colleagues were 3 insertions or deletions. And let's see what 3 insertions or 3 deletions do to the message and the polypeptide. So if I have CAU and then the first C is deleted, that creates a mutant, but then we again delete another A. Then we have U, CA, and let's say we delete the U after this. And then CAU, and so on. This would result in a message which is CAU, AUC, UCA, but after this third amino acid, the message would revert back to CAU, and so on. And therefore, when you have 3 deletions, and similar logic applies for 3 insertions, just like the suppressor mutations, where you reverted back to the wild type sequence of the amino acid sequence of the polypeptide, 3 insertions or 3 deletions cause a similar reversion. And this is only possible if the codon were 3 base pairs long, exactly 3 base pairs long, since if the codon were 4 base pairs long, let's say, then 3 deletions or 3 insertions, or a combination of 3 insertions and deletions, would still cause a frame shift mutation. Therefore, the analysis of these mutants of T4 phage allowed Crick and colleagues to prove that one codon is 3 base pairs or 3 nucleotides long.